All right. In this next video, I want to spend some time talking about leukocytes and the white blood cells of the bloodstream. So, on that note, let's just get rolling. Leukocytes, bear in mind, are good way to think of leukocytes or white blood cells is that they essentially are our immune system. Okay, these carry out, the bulk of the responses and effects that our body undergoes when we are fighting off a foreign invader, like an allergen or a parasite or a virus, a bacteria, basically pathogens. Okay, and white blood cell counts tend to run much lower. We said that men typically have, around 4 to 5 million cells per microliter, women about 3.8 to a little over 4 cells per microliter of blood million, whereas most adults have about 5 to 10,000 white blood cells per microliter, significantly smaller number of cells in proportion. And there are reasons for that. One is because, when we synthesize, these cells are synthesized in red bone marrow, just like red blood cells, but most of these are also stored in the marrow. Okay, the red marrow, until we need them. Okay, so if we get an infection or there's tissue damage, and then that tissue becomes damaged, and then it may release these chemicals into the bloodstream, which will diffuse into the red bone marrow, which will cause the release of these into those big sinusoid capillaries of the red bone marrow. Okay. Also, there's a lot of white blood cells that are stored within lymphatic tissues. Not just stored, but also produced. Lymphocytes are, produced and stored in here. All right, and these particular cells are responsible for carrying out the actual, specific immune response, responding to antigens. All right, which I'll talk about later on. But so these cells are primarily found in the, within lymphatic tissues, lymph glands, lymph nodes, patches, and so on. And then, a lot of the phagocytes are, are produced and then migrate into the bloodstream, typically on their way to other tissues. So you get the gist here that white blood cells, with the exception of these lymphocytes, are produced in red bone marrow, but the ones that you find in the blood are more than likely on their way to another tissue somewhere to be stored. Okay, so circulating white blood cells are typically on their way to another destination in the body. Now, unlike red blood cells, white blood cells have a conspicuous nucleus. That is one of the biggest key, that is probably the biggest key factor when you're trying to determine the appearance of a leukocyte. Okay, you look at the shape of the nucleus. All right. So keep that in mind. And also you're going to look for how granulated the white blood cells are, because essentially they have a lot of granules, which are basically storage vesicles, these storage pockets of excretions that they release, or secretions that they release, that are going to basically be used in the immune response, like a chemical signal or something that's designed to kill a bacteria or virus or so on. All right, so how granular are they? And then, what is the shape of their nucleus? That's how you tell the different white blood cells apart. All right, and then when looking at the different classes of white blood cells, we can break these down into 2 distinct classes. One of them would be the granulocytes. The other would be the agranulocytes. Now, agranulocytes are not truly without granules. It's just when they first started describing these, they were just hard to notice until we were, got stronger scanning equipment and were able to look at these cells more in depth. But or in the early days of studying blood under the microscope, when they were staining these, they didn't look granular, so they called these agranulocytes. Okay, now granulocytes are going to be, a lot more noticeable, because the granules are going to be very easy to spot within their cytoplasm. Plus, yeah, so I shouldn't say plus. So now, what's really important about understanding white blood cells is not just that, we mentioned that number before. We said that there are about 5 to 10,000 cells per microliter of blood. Per cells per microliter of blood. All right, that's important to know, Because if these numbers go up or down, that could indicate if a person has an infection. It could indicate if a person has a bone marrow problem. It could indicate if a person lacks an immune system, is going to be susceptible to an infection. But what's also important is to look at the differential. Is to not only count the number of cells, but to do a differential count. When I say a differential count, which white blood cells are going to be found in more abundantly than others? Okay. So in normal levels, there's usually about 62% of the white blood cells you see in the circulation are neutrophils. About 2.3 are eosinophils. 0.4% are basophils. Okay. 30% lymphocytes. And a little over 5 would be monocytes. You add all that up, that comes out to 100%. All right. Now, each of these different types of white blood cells are responsible for attacking or carrying out different tasks, okay, or attacking, different invaders or pathogens. For example, neutrophils are what are called first responders. Okay. These are what are called first responders, and typically go after bacteria. Like if you get a cut in your skin, neutrophils are going to invade that area like crazy. All right, eosinophils typically attack parasites. Okay, basophils are commonly used in allergic reactions. Okay, lymphocytes typically go after, cancer cells. Viral infections, basically bad cells. Okay, monocytes are phagocytes. They're going to play a very key role in the inflammation process and tissue damage. Okay, so for example, if you if you understand these basic numbers, and let's say you're whether you're working in a lab or you're looking at someone's lab chart, and you see this person, their basophils went from 0.4 up to 5%, you're going to think maybe they're having some kind of systemic allergic reaction. Or maybe there's a bacterial infection if neutrophils are at 75%. You get the picture here. Okay, so it's important to not only know the specific numbers, but do a differential count with this. Now, when it comes to telling these cells apart, you look at the morphologic characteristics. Neutrophils have a nucleus that has 3 to 5 lobes to them, so they're multilobular. And the granules are typically very fine. They're noticeable, but they're not very big. Okay, the cytoplasm is going to look very grainy in appearance. Okay. Eosinophils have a bilobe nucleus that's shaped kind of like so. And the granules are going to be very big, and when it says rosy, kind of red in color. All right. Basophils have these cells have an S-shaped type nucleus, and they have enormous granules inside of them. Okay, very large granules. They're so big, it's going to be hard to see them. Heck, if you even find one of these in a normal person's blood, those are tough to find. All right. Lymphocytes I always thought neutrophils, because of their multi-lobe nucleus, are easy to spot. And I always think lymphocytes are also the easiest ones to spot, because basically, let's say here's the cell, their nucleus takes up almost the entire surface area of the cell. Okay, they have a very large conspicuous nucleus. These are cells that when there's an infection or there's something wrong, when we need to utilize these are cells that are going to start to divide a lot. These are cells that are going to manufacture a lot of material that they're going to secrete out of themselves, like antibodies and other chemical messages that coordinate the immune response. Okay, depending on whether it's a T or a B lymphocyte. All right. Monocytes are also relatively easy to spot because their nucleus is horseshoe shaped. Okay, it's pretty big, and like I said, it is shaped like a horseshoe. All right. So, like I said, these cells are pretty easy to tell apart. When you take a look at them. So let's take a look at some examples. But before we do this, let's just review the functions. Remember neutrophils, bacteria, first responder cells. Okay, Eosinophils, parasites. Basophils, allergies. Lymphocytes, viral, cancer, basically bad cells, antigens, foreign antigens. And monocytes, these are phagocytes. Okay, we're going to need to utilize and mobilize these a lot during inflammation. So basically when any one of these are possibly going on. Okay, so these are the functions. So remember, these are the functions of these cells. Now let's take a look at some specific examples of these. So a neutrophil. Again, bear in mind, when you're taking a blood sample and you're looking at these, you're going to have to weed through a lot of red blood cells. You just know that there are far more erythrocytes in peripheral blood than there are anything else, including platelets. All right. Now you'll notice as you take a look at this picture here, as you take a look at the this neutrophil, notice that you see, you know, it has 4 lobes to its nucleus. All right, and then you can see the granules. Okay, you know, they're very fine granules within the cytoplasm of that cell. All right, like I said, these cells typically have a very grainy looking appearance to them with these multi-lobe nuclei. All right, and typically neutrophils, as I mentioned before, what they do is they, you know, like for example, let's say here's your skin. You get a cut in your skin. All right. And then you get some bacteria leaking in there. That damaged skin then is going to leak out some chemical messages that are going to go to the red bone marrow. Let's abbreviate that. They're going to go to the bone marrow, which then that's going to stimulate the bone marrow to dump out the neutrophils into the bloodstream, which are then going to migrate to this damaged tissue. And then what's going to happen are there's going to be some mast cells around the connective tissue spaces here, and these mast cells are going to dump out the chemical histamine. Okay, some basophils may be doing this as well, and now the mast cells, I shouldn't say basophils, ignore that. Okay, and then histamine is going to stimulate the pores to retract, or the cells to retract and make the pores bigger in capillaries, and then what's going to happen are these neutrophils are going to squeeze through into the tissue and attack the bacteria. And so they're going to degranulate, dump out a bunch of enzymes, and those enzymes are going to basically kill the bacteria. Plus, neutrophils are also phagocytes. Okay, so they'll not only, damage they'll damage some of the healthy cells and tissues in there too while they're dumping out all those enzymes, but they'll also phagocytize bacteria. Okay, and I forgot to mention this process of squeezing out of here is called diapedesis. Okay, diapedesis. All right. And then the neutrophils are going to attack, and eventually they're going to die. You know, neutrophils are only going to live for maybe a couple of days at most. But if we're really mobilizing these and they're attacking pretty heavily, then they may only live for a few hours. Okay, but those are essentially neutrophils and what they do and how they work, and that's kind of a little bit of a basic intro into the process of inflammation as well, which you'll learn about later on. All right, granulocytes are cells that are granulocytes, bonehead. Eosinophils are granulocytes, but eosinophils, remember we said that these have these very, very large, rosy granules. Whoops. Large rosy granules, okay? And then these have that kind of bilobe shaped nucleus. You can kind of barely see it in there. All right, and then when these degranulate, these are, again, these release chemicals that are responsible for killing parasites. Okay, I mean, when I think parasite, I'm typically thinking worms. Okay, so there are certain parasite infections, especially worm infections that you get that you tend to see more in underdeveloped third world type countries. I mean, I'm not saying people in developed countries don't get parasite infections; they're just not as common. But then if you get one of those invaders, then you'll mobilize more eosinophils. Now, bear in mind that worms are typically going to be too big to phagocytize, so eosinophils really don't act as phagocytes. But what they do, like I mentioned, they just dump out a bunch of these chemicals, which will, cause, if not the death, at least a lot of damage to those worms, which will eventually make them die. And then, phagocytes will clean up the mess from there. All right, so that's eosinophils, and then we've got basophils. Again, you can kind of make out the nucleus in this picture here, but barely. Again, these also have very large, thick granules that store two main chemicals, histamine and heparin. They also do release some serotonin. All right. And you guys all know what heparin does. Heparin prevents the formation of clots. Okay, it prevents the formation of a clot. Histamine basically is a chemical that that makes capillaries open up more. So basically that increases capillary pores, this diameter. All right, and serotonin is in peripheral circulation, is responsible for vasoconstriction. Vasoconstriction. All right, so now typically these cells are, these cells along with some mast cells can interact with the antibody IgE. Okay, immunoglobulin E, and IgE is what's responsible for being mediated with allergic reactions. And if these antibodies are interacting with these cells, then we would say these are sensitized by the antibody. And then when you come in contact with that allergen or that specific antigen again, then that will cause these cells to degranulate and dump out histamine. And then basically you will see the effects of inflammation, because that's essentially what histamine promotes, is inflammation. And with inflammation, you need increased blood flow to a particular area, and that's where the heparin comes in. Okay, you dump out heparin, that's gonna slow down the clotting process. It'll be easier to deliver blood to that area. All right, serotonin is definitely handy for whenever you've got damaged vessels and you need that vessel to constrict to minimize blood flow to the area. All right, so those are basophils, but bear in mind that, again, these are by far the least abundant of the leukocytes that you're going to find. About, again, barely 0.5% of peripheral white blood cells are going to be basophils. And then the agranulocytes, now again, truly these are not truly agranulocytes. You can see kind of the grainy looking appearance in the cytoplasm, but notice that lymphocytes have that large nucleus. They have that large conspicuous nucleus, okay? And as I mentioned, the nucleus takes up almost the entire size of the cell itself. It's very big, okay? Very, very big. Now, the nucleus, or like I said before, these are cells that, right now, I honestly can't look at the cell and tell you, is this a T or a B cell? Okay, T or B cells, before they become active, are pretty hard to tell apart. Okay, but then once we come in contact with a, at least under a microscope anyways, they're hard to tell apart. But when we come in contact with a specific antigen and then we coordinate, these are responsible for what's called the third line of defense. Okay, or we can call that specific immunity. Specific immunity. Basically, these are cells that respond to antigens. Okay, so these attack very specific parts of foreign invaders. Okay, well, they attack foreign invaders based on how they identify them, by their antigens. Okay, so basically, when these cells are activated, they either become, basically they are B cells that form into plasma cells, which are basically all nucleus and rough endoplasmic reticulum, and then they just sit there and pump out antibodies into the bloodstream. Or the T cells start to divide, and then they become T helper cells. They become cytotoxic T cells, or they become T memory cells. Okay, oops, TC, TK. Okay, Helper T cells, release a lot of chemicals that are very important for coordinating the immune response. Okay, without T helper cells, this immunity is going to be almost non-existent. That's what happens with people that have AIDS. Okay, the HIV virus affects all the white blood cells, but it's the T helper cells that, when those cells start to decline, that's when you see the big problems with the compromised immune system. Cytotoxic, or killer T cells is pretty straightforward. They're TC. Cytotoxic T cells is pretty straightforward on what those do. They kill. Okay, and then these memory T cells are basically hanging out, and they're genetically programmed. So if we ever encounter that antigen again, boom, we'll be able to just attack it very effectively. B cells also do that as well, form memory B cells. Okay. So lymphocytes are very important for specific immunity. And then, again, you could see some of the granules in here, but these are monocytes, and remember that these are phagocytes. And as I mentioned before, you can see the horseshoe-shaped nucleus is pretty obvious. And again, these are typically on their way to another tissue. And then these will more than likely migrate into other tissues of the body. If they migrate into connective tissue, then we call them macrophages. Okay, if they migrate in the liver, we call them Kupffer cells. If they migrate into the, if you find their way into the skin, they're dendritic cells. The brain, microglia. Okay, you get the picture here. So basically, these cells, they're still, even though they have different names, they still all do the same thing, but they all look differently. These cells adapt to the environments that you find them in. Okay, so even though they are phagocytes, they're all going to look very different under a microscope than what you see here in front of you. All right, and so again, these are phagocytes that are, in the circulation that are more than likely on their way to migrate into a tissue to undergo the inflammation response, whatever the process need be. All right. Now, what I'd like for you to do is to take a second and to take a look at this picture and then identify the cells. What do you identify every possible cell you can? What do you see here? I'll give you a second to do that. See if you can spot stuff. Or pause or do whatever. Okay. So I'm going to move. I'm not going to give you guys the answers because I want you guys to make sure that you're doing this right. Now, leukopoiesis, this is a, gosh dang it. Leukopoiesis then is a lot like erythropoiesis, the production of red blood cells, except we're obviously producing different cells. Now, this is a process that is regulated by these, CFUs. I don't know why I put stimulating. Sorry about that. Forming units. Okay. Now, again, this is regulated by other white blood cells or other physiologic mechanisms. For example, as I mentioned before, if we get sick and there's tissue damage, damaged tissues may dump out these CFUs. They'll go to the bone marrow, and which will enhance or stimulate the production of these, or the release of these white blood cells. Okay, some lymphatic organs may be releasing these chemicals. How these are regulated varies, is pretty variable within the body. All right, but that's, essentially leukopoiesis, the production of this. And typically when this happens, what you're going to notice, what was I going to say, after these cells are released into the bloodstream, okay, they're produced in the bone marrow or the lymphatic organs, and they're released into the bloodstream, and then they migrate their way to other tissues based on what are called chemotactic factors. Okay, and remember that these cells, when we produce them in the bone marrow or lymphatic tissue, start out as stem cells as well, and then based on the type of CFU that it comes in contact with, then it may differentiate into, these may differentiate into, again, through a step-by-step process into a monocyte. Okay, they could differentiate into a neutrophil. Okay, could differentiate into an eosinophil. You get the picture. Okay, so if it's CFU, neutrophil, yeah, it's going to stimulate the production of a neutrophil. It's CFU, you get, okay, you see how this works? All right. Whereas these lymphocytes, this process takes place within lymphatic tissues, lymphatic organs, and then they go through a step-by-step division process where they will eventually become mature lymphocytes and kind of hang out within those tissues. And then last but not least, let's talk a little bit about leukocyte disorders. For example, if a person has leukopenia, then what's going to happen is, that means, remember, penia means thinning. Okay, leukopenia. Now, this is caused by, essentially damage to bone marrow, where you damage those stored white blood cells, where you damage those stem cells, various poisons and diseases, and so on. But essentially, what happens when you have a low white blood cell count below 5,000 microliters, now you have an elevated risk of infection. Okay, and then when you have an elevated risk of infection, then, you're going to get sick a lot more. And if it gets bad enough, you could get what are called opportunistic infections. These are infections that people don't normally get, okay, if you have a healthy immune system, okay? Like Kaposi sarcoma, fungal infections, all right? And and so on. You don't get internal fungal infections if you have a normal working immune system. We kill it. It's as easy. Fungi are as easy to kill. Okay, the Kaposi sarcoma caused by one of the herpes viruses. Okay, we kill that relatively easily. But without an immune system, these invaders that we're normally exposed to, that we also easily handle, can now handle us. All right. So that's what happens when you have a low white blood cell count. Leukocytosis then would be when you have a blood cell count over 10,000 per microliter, and that usually is indicating that a person either has infection, allergy, or disease. All right, and then that's why you do a differential count, because you need to establish what kind of infection. Are they having an allergy? What kind of disease is taking place? So remember that these different white blood cells have different functions. We're going to produce and mobilize white blood cells depending on what's going on with us. Okay, and that's why you remember that most white blood cells are stored in bone marrow and other tissues. That's why the count is going to go dramatically up in the blood, because they're going to be dumped into the bloodstream, or we're going to be utilizing them. And then they're going to die, and then we'll have to make more new ones all over again. Okay, and then last but not least, leukemia. This is a cancer of hematopoietic tissue. This is essentially, overproduction of white blood cells. All right, and there's various forms of leukemia. I'm not going to sit here and go over every single one. Some are, I don't want to say they're not very harmful, but some are more easy to manage and treat, whereas others are extremely dangerous and life-threatening. But either way, this is not a good disease. So not that any disease is. All right, but what happens then is when people get leukemia, basically remember, all of our blood cells start out as those pluripotent hematopoietic stem cells. Sorry, I had a brain fart there. They all start out as the basic same type of a stem cell. And then what happens then, if for whatever reason these stem cells are just constantly just being manufactured into white blood cells, a person's going to have a lower, red blood cell count. Person's going to have a lower platelet count. So then as a result, they're going to have a harder time utilizing oxygen. They're going to have a harder time clotting and, preventing bleeding if they get cut. And oftentimes leukemia is a pretty silent disease. It doesn't present with symptoms very well. And, like for example, if I'm an adult and I have leukemia, I may figure out I have it because I hack myself shaving. And then, I'm not supposed to just bleed all day after that. I go in and get it checked out. They run a blood test and they see this going on. Okay, or you may get really tired, fatigued really easily. You get the picture. All right. So these are some problems that can go with leukocytes, in terms of their production. And like leukocytosis isn't necessarily a bad thing, because we want to increase our ability to fight off anything that's invading us. Okay. In the next video, I will be talking about platelets and hemostasis.